So today I'm going to talk to you about cancer dormancy and give you an extracellular matrix perspective. It's going to be an overview of the work I've done during my postdoc. And first to start, just like a general idea of cancer progression. So we know that cancer is one of the leading causes of death worldwide. And 90% of this cancer-related death are due to metastasis. So far, metastatic disease remain incurable. And metastasis rise from the cells that leave the primary tumor, they disseminate, they extravasate, and they end up in different part of the body where they will grow and cancer will spread. A long time ago, Stephen Paget was studying breast cancer patients, and it was looking at the organs that were colonized by metastasis. And he found out that metastases are not developed randomly. And he postulated the seed and soil theory, where metastasis will only develop when the seed, the cancer cells, and the soil, the microenvironment, are compatible. So in other way, take a seed, offer a different soil, it will only grow if the soil allow the growth of these cells, of this seed. But if you take the idea of the seed a bit further, we know that certain seed have the ability to delay this germination and wait for proper condition to grow. And this is what we call cancer dormancy. So this is an old concept from the 50s, where Hadfield has first proposed that some cancer cells could remain in tissue for a very long time without dividing, but will keep this capacity to proliferate at some point. So the cancer dormancy happened during what we call the minimal residual disease. So you had a patient that had a primary tumor, the tumor was treated very often like resected by surgery, treated with chemotherapy. And then we enter this minimal residual disease when the patient is in remission and we are like in the wait for metastasis to occur. So if we take back the idea of the seed and the soil, the seed has disseminated. In this first step, the cancer cells encounter a microenvironment that's unknown, and it's going to enter into what we call a cellular dormancy, where the cells is sitting in G0, not developing and adapting to this new microenvironment. At some point, and we don't know yet exactly how, the cells will be able to reactivate. And then we'll enter into a phase of tumor mass dormancy, where the lack of angiogenesis and the immune system surveillance will not allow the cells to grow to a certain point, and will be in a state where there is an equal amount of growth and death, and this will still be under the clinical threshold. But if the cancer cells are able to resolve this problem, they will reactivate and actively grow metastasis. So cancer dormancy was found in multiple cancer types, and in different organs, such as the bone marrow, the blood, the lymph node, the liver, and the lungs. But as much as metastases were not found random, all the microenvironments are also not equal when it's regarding dormancy. So the early work of Roluaguirre-Guizot showed that there is a permissive and restrictive microenvironment that will time the dormancy period. In other terms, some microenvironments are long-term dormancy, such as the bone marrow and the liver. Cells that will end up in the liver and the bone marrow will have longer dormancy period, compared to short dormancy in permissive microenvironment, such as the lung. So what's this soil that we call the tumor microenvironment? It's made of a cellular component made of stromal cells mainly, and tumor cells, and an acellular component made of soluble factor and the extracellular matrix. And this last is the one I'm going to concentrate right now for the rest of the talk. And if you look at cancer progression, the cells will encounter a lot of different matrix microenvironment from the rise of the primary tumor to the endpoint of the metastasis. First, in the core of the primary tumor, the extracellular matrix, the cells will be exposed to a certain type of matrix. Then when the cells will invade the microenvironment, we will have a different type of matrices here. Once ending up in the distant site, in the dormant microenvironment, the cells will be exposed to another type of matrix. While when the cells will regrow, will certainly be exposed to a different type of matrix as well. So how do we study the extracellular matrix in the lab? It's by what I call catching the free signal using two-photon imaging. So two-photon imaging increases the spatial resolution. It allows deep imaging of solid tissue, and it generates the second harmonic signal that we use to see organized matter. So basically, when the laser hits what we call an optically nonlinear medium, such as like an organized matter, like the fibrillar collagen, they will generate like a delay in the wavelength, and you will be able to see without any staining, just by opening the mouse, the extracellular matrix. And here is an image of the mouse skin in second harmonic, where you can see the junction between the skin matrix and the muscle fibers. If we use a software, we can color code it and extract quantitative numbers to measure this matrix organization and alignment. So this is important to look at matrices, because regarding where the cancer cell is going to end up, you have a very different type of matrix organization. Here are shown the brain, the lung, the liver, and the spleen matrices of a mouse in second harmonic. And this is important because the early work of Patricia Keeley has shown that in breast cancer, there is an alignment of the matrix that correlates with the cancer progression and correlate with poor outcome in cancer patients. But now we're able to go a bit further, and we can do ECM-enriched proteomic of these different organs and get access to what are the different components of the extracellular matrix in these different organs. And this is the work of Alexandra Naba during a postdoc, where she described the matrisome. The matrisome is a signature of more than 1,000 proteins that are making the extracellular matrix. It's composed of the core matrisome, where you will find ECM glycoprotein, collagens, and proteoglycan, as well as associated proteins such as secreted factors, ECM regulators, and ECM-related proteins. It is well known now that extracellular matrix is important in cancer progression, and it has been shown through its composition, its organization, its stiffness, and the receptor available that can control cancer progression. But what about ECM and cancer dormancy? So when I started my postdoc, we knew about ECM composition, that putting cancer cells in a basement membrane-like gel in 3D would promote a dormant-like state, as well as the thrombosponding derived from endothelium will be able to induce dormancy of cancer cells. It was also known that the stiffness could also influence the dormant state, where stiffer fibrin gel and softer collagen gel will restrict the tumor growth. And about the receptor involved, the work from Rogolio Aguirre-Guizzo showed that a loss of integrin function will prevent dormant cells to reactivate the proliferative pathway downstream of integrins. And nothing much was known about the ECM organization. So what I was interested in was to understand the role of ECM in this cancer dormant state. And in other term, like, do tumor cells control their metastatic fate by sensing and regulating ECM composition and architecture in this distant site? And in other term, what's the matrix different in this around these dormant cells compared to this proliferative tumor? We'll see in this talk that the organization, the composition, and the receptor involved will be described for this dormant state. So in order to study dormancy, we need a dormancy model. And for that, we have a human head and neck squamous cell carcinoma model, where the T-HEP is actually a PDX for patient that we maintain in chicken embryo and pass every week. And there they retain their ability to grow a tumor in animals. If you take those T-HEPs and you put them in culture for more than 40 times, you will end up with a dormant clone that we call D-HEP. As a result, if you inject the T-HEP and the D-HEP in the mouse, you will have a delay of the growth of the D-HEP, allowing us to have this dormancy period where we can study the dormant state. We also used 2 murine breast cancer models where D2A1 and 41 are proliferative clone, and D2OR and 4007 are dormant clone, in the same way that they are not proliferating in the distant side, while this one for metastasis. So the first thing we did is to use a CDK2 sensor created by Sabrina Spencer during a postdoc to be able to visualize by microscopy what was the cell cycle state of the cells that we will be able to image with the 2 photon. And what we did was to grow a T-HEP tumor, image this tumor, then resect the tumor by surgery, wait for 4 weeks, and observe the matrix. And here's what we found. In the primary tumor, no surprise, the extracellular matrix is aligned and the cells are growing. Here, the cells also express a H2BRFP to allow us to distinguish nucleus from cytoplasm in those cells. Then 4 weeks after surgery, in the local surgery site and in the distant site, like the lung, we found these 2 very interesting things. Each time that the cells will be single and concentrating the fluorescence in the nucleus, like a G0 cell, we'll find a curly matrix in the local site and in the distant site. And when the cells are proliferating and forming micro and macro metastasis, the matrix will be aligned. We grew autotopically these models and found this and quantified this matrix architecture and found this difference back between T-HEP and D-HEP, where D-HEP have matrix are way curlier than T-HEP. And that was reproduced in the 2 model of murine breast cancer, where D2A1 and 41 had a line matrix compared to D2OR and 4007 matrices. We were able to quantify this using an ImageJ plugin called Orientation J. And here you can see that how you read this graph is like the highest is the peak, the more aligned is the matrix. So you can see that all the proliferative tumors have higher organization and higher linearization of the matrix. So we took this T-HEP and D-HEP nodules and did RNA sequencing and found a dysregulation of the NABA matrisome signature. We then collaborated with Alexandra Naba and did the ECM-enriched proteomic of these nodules and tumors and found an enrichment in collagens in the dormant cells. We validated this enrichment doing picrocirrus red, and you can see more red staining in the D-HEP compared to the T-HEPs. And because we use a human cancer cell into a nude mouse, we were able to dissociate human peptide from mouse peptide. And doing this, we were able to access the tumor-derived matrix coming from the cancer cells, and the stroma-derived matrix coming from mainly the fibroblasts in the mouse. And doing this for the collagen family, we found an enrichment in collagen 3 in the tumor-derived ECM. This was the only collagen enriched in the dormant nodule compared to the proliferative tumor. And we also found this enrichment into the stroma-derived ECM. We confirmed this by staining collagen 3 in the tumor and in the nodule. And if we go a bit further into the experiment I showed you before and section the lungs of the T-HEP mice and compare single cells and proliferative micrometastasis, we found an enrichment of collagen 3 around the single dormant cells compared to the micrometastasis proliferative, like we had the K67 staining showing that those proliferate. We analyzed cancer patient tumors. We stained them for collagen 3 and found that that's head and neck squamous cell carcinoma, by the way. We found that the patient that have no lymph node, positive lymph node at the time of the cancer detection, compared to patient that had a positive lymph node, that's a worse outcome for patient. We found an enrichment in collagen 3 in the good prognosis for this patient. And that suggests that collagen 3 could be used as a marker for poor outcome, or I mean, at least this accumulation will show a better outcome for patients. So we were very much interested to know if this collagen 3, and could we trigger dormancy by enriching the microenvironment in collagen 3? So in order to do that, we co-injected proliferative T-HEPs in DPBS in one flank of the mouse, and those T-HEP in a solution of collagen 3 on the on the left flank of the mouse. And here's what we found. We, of course, injected the same number of cells, and what we found is that the side that was injected with collagen 3 it decreases in size, so the proliferation was lower in the collagen 3 enriched condition. This correlates with a decrease in phosphohistone 3 staining, that's a proliferation marker, and an increase in the positive P27 nucleus that's used as a dormancy marker. If we lay down the T-HEP on collagen 1 and collagen 3 and count the number of mitosis over 18 hours, we also found a decrease in mitosis when the cells encounter collagen 3. So this showed us that the collagen 3 can induce dormancy of proliferative cells. But what about after surgery? Could this be used to prevent local relapses? So in order to test that, we grew T-HEPs in 2 groups of mice. And when the tumor reaches a certain size, we do the surgery. And we either treat the mouse with a DPBS loaded sponge. It's a dental sponge made of gelatin, or a sponge loaded with collagen 3. So this is the tumor grow before surgery, and then we monitor for local relapses. And what we found is that in 80% of the case, the mice that were treated with the controlled sponge had local recurrences, while only one animal treated with collagen 3 had a local recurrence. So with that, I hope I show you that type 3 collagen can induce cellular dormancy. And when the cells disseminate to a distant site, if they sense collagen 3 in the microenvironment, they will be able to start this dormancy program. But we know that the tumor-derived ECM, it's very little compared to the stroma-derived ECM. And in the head of people, it's always like fibroblasts that make up most of the matrix. In fact, in our ECM-enriched proteomic, we found only 6% of the peptide coming from the tumor-derived ECM compared to the stroma-derived ECM. And if you recall the result from before, collagen 3 was upregulated in both compartments. So we're very much interested to understand what was the role from the stroma-derived and the tumor-derived matrices. And for that, we use the CAM assay. The CAM assay is used since the 80s to study metastasis, and it's a very good model because it's fast and affordable. And all the results I will show you with the CAM assay is translated in mouse in the paper. So how does that work? At day 10 of the development of the embryo, we inject the cancer cells. 6 days later, we collect the tumor. So a dormant nodule versus a proliferative tumor at same scale, for you to see the differences. We can collagenase those tumors and count the number of cells. And then after counting, if we have approximately the same number of cells as day one, the condition will be considered dormant. If we see an increase, it will be proliferative. And if we see a decrease, we have a condition killing the cells. Those tumors can also be used for RNA protein extraction and do all the molecular biology that can be done with it. But I want to show you, before going back to the paper, I just want to show you some cool thing you can do with the CAM. If you put the egg under the 2-photon imager and you inject dextran in the blood vessels, you can see the organization of the tumor in a T-HEP tumor where the cells express GFP, and in a D-HEP nodule where here you can see cells sitting sparse compared to this packed tumor. So these are the difference you can see using the microscope. You can also organize the time lapse and see the cells moving and forming protrusion over time. And you can also mimic a tail vein assay by injecting the cells directly in the vein, and a few days later, collecting the organ. You can find the GFP cells. For instance, here's the T-HEP going to the brain, the liver, and the bone marrow. But going back to the paper and being interested in understanding the role of the stromal and the tumor-derived microenvironments, matrices. So what we did here was we co-culture fibroblast control or depleted for collagen 3 with dormant cells expressing the CDK2 sensor I showed you before. So we imaged this different condition, the control condition, and 2 different SI in the fibroblast, with the idea that the dormant cells here will be exposed to a microenvironment that's less enriched in collagen 3 compared to the control. And we quantified the different cells in the different state of those cells. And we found no differences between the control and the cells exposed to fibroblasts depleted for collagen 3 between the different phases of the cell cycle. And this was able to tell us that as much as a proliferative cells encountering collagen 3, extracellular collagen 3 will trigger dormancy, once a cell is dormant, the stromal collagen is no longer necessary to sustain this dormant state. So how do they sustain this dormant state? What we knew is that from the RNA sequencing is that dormant cells overexpress collagen 3 at their RNA level compared to proliferative cells. And here, if we deplete collagen 3 from the dormant cells, we see the cells regrowing. So this is in the CAM tissue, and we did it with tail vein injection. And as soon as 5 days after injection, we can start seeing proliferation. And here we quantified it and we saw an enrichment in cells in the S phase and the G2 phase when the dormant cells are downregulated for collagen 3. We did the reverse, where we forced proliferative cells to overexpress their own collagen 3 with a plasmid that overexpressed collagen 3 upon doxycycline treatment. And here we saw a decrease of the tumor size in the CAM, and we also saw a decrease in the phosphorylation of histone 3. That's a proliferation marker. And associated to this, we saw a change in the matrix organization. Cells that were depleted for collagen 3 will align the matrix while they are growing. So this is the second message I have for you. So once the cells are disseminated, senses the stromal-derived collagen 3 and enter in this dormant state, the cells is now relying on their own tumor-derived collagen 3 to sustain their dormant state. So the next question was, which receptor will sense this tumor-derived collagen 3? And there is 2 families of receptors that are ubiquitously expressed that can sense collagen, that are the integrins and DDRs. We went back to our RNA-seq and look at all the receptors that were dysregulated between THAP and DHAPs. And we also knew at the time that DDR1 was part of the ER positive and negative breast cancer cell line dormancy signature from the work of Julio Aguirre Guizzo. We did a egg screening. So using the CAM, we downregulated each of these receptors and look at tumor growth. And we found for DDR1, for integrin alpha 1 and for integrin alpha 10, the downregulation of these receptors will allow the cells to regrow. We focused on DDR1 because it was the highest effect of those three. But I just want to point out that not only DDR1 depletion allowed the cells to regrow, and there is somewhere, somehow, an involvement of the integrin as well. We did a Western blot for the different model we had and found an accumulation of DDR1 in the DHAP compared to the THAP and in the D2R compared to the D2A1. So if we deplete DDR1, inject them in tail vein, and look at the lungs 3 weeks later, we found that in the depleted conditions, we have micrometastasis in the lung, and the matrix is aligned compared to the dormant condition. And in 60% of the case, we saw macrometastasis in these lungs. So DDR1 is a transmembrane receptor that has a discretin domain that can bind collagen and a kinase domain that can trigger signal, intracellular signals. We have a binding deficient mutant and a kinase dead mutant that we use for a rescue experiment. And here are the results. So those are CAM assay. These are the DHAP that are dormant. If you downregulate DDR1, they regrow. If you downregulate DDR1 and express an empty plasmid, they keep growing. If you overexpress full-length DDR1, you rescue the dormant phenotype, as well as if you overexpress a kinase dead mutant. However, if you overexpress a binding deficient mutant, the cells keep growing. So, and that suggested that the dormant phenotype that we saw was due to the binding of DDR1 to collagen 3. And that matches our adhesion assay to collagen 3, where the dormant cells can attach the collagen 3 matrix. When the cells are depleted for DDR1, they lose this adhesion. And if you rescue the phenotype of overexpressing DDR1, you also rescue the adhesion to collagen. And to test the result of the kinase dead, we treated the cells with nilotinib that inhibit the phosphorylation of DDR1, and found that when the cells are treated with nilotinib, they remain dormant. So the kinase domain of this receptor is not involved in this dormancy pathway, but the binding to the collagen 3 is. And this is the third message where DDR1 is necessary to sustain the cellular dormancy, and DDR1 will bind collagen 3 and sustain this dormant state. And something interesting to us was that as the cells were regrow, they will align the matrix as well. And we perform ECM-enriched proteomic of this controlled DHAP and depleted for DDR1, and found a decrease in the abundance of collagen 3 in the depleted cells that was confirmed by staining. Here, the regrowing cells has a downregulation of the expression of collagen 3. And that downregulation is happening at the RNA level. So if you compare THAP controlled and DHAP depleted for DDR1, we found a significant decrease in expression of collagen 3. So we went on and searched for a transcription factor that could regulate the collagen 3 expression to sustain the dormant state. We went back to our RNA-seq and had 3 targets that are STAT1, FOSB, and RF7. And only the depletion of STAT1 was able to show a decrease in the expression of collagen 3 at the RNA level. If you downregulate STAT1 in the T-HEP, the cell will regrow in the CAM, and they will also downregulate DDR1 that suggested that there is a feedback loop between DDR1, STAT1, and collagen 3. If you perform tail-vein injection and look at the lungs, we also found micrometastasis in the lung of these animals compared to the control. When laid down on collagen 3, we found an accumulation of phospho-STAT1 in the nucleus of the cells that was significantly enriched in the nucleus of the cells, suggesting that there they will actively transcribe collagen 3 to sustain the dormant state. So with all this, that brings me to the conclusion where we use an imaging approach of the dormant state, focusing on the extracellular matrix. We pair that with a quantitative proteomic approach to describe the microenvironment ECM of the dormant state, and uncouple a new mechanisms that show dormancy driven by an ECM component that is collagen 3. So here's how we think it works. Cells leaving the primary tumor will end up in a distant site. There they will sense the soil ECM made from the stroma. And if they encounter collagen 3, they will enter in this dormant state. They will first bind this collagen 3 to DDR1. They will start their dormant program, upregulate collagen 3, secrete the seed ECM that's enriched in collagen 3. And this seed ECM will allow the cells to sustain their dormancy, but also instruct the stroma to make more collagen 3. And all this is accompanied by an ECM architecture switch where dormant cells are surrounded by a curly matrix and proliferative cells are surrounded by an aligned matrix. And this curly matrix made of collagen 3 will bind DDR1, activate STAT1, create more collagen 3 to lock the cells in this dormant state. So with all that, I would like to thank you for your attention. Thanks the people in the lab, all the former students that helped through the years, our collaborators that give us precious help on the study, and our findings. The last thing I wanted to show you is something we started during the pandemic. So if you're interested in the extracellular matrix and you want to join our community, in 2020, we created what we call the Meshwork. It's a community of early career researchers working on the extracellular matrix. We organize meetings. We have a Slack platform where we can ask questions and share papers. We've been now reaching something like 300 members. We have a lot of PIs that were involved helping us. So if you want to follow us on Twitter, it's the at the Meshwork 2020. And if you want to join, please email us and we will make you join the Slack platform. Thank you.
Thanks a lot, Julie. It was very nice. So it's time for questions. Do not hesitate to raise your hand, turn on your camera and ask questions. Yes, Isabelle?
That was great. Super. Thanks. I love it. So I have a question regarding the selection of your DEP cells. So to my understanding, in your diapositive, you showed that you're passaging cells until the 40th passage to get them. And then, so you re-inject them. So you count the same number of cells and you re-inject, and then you score what? You score the size of the tumor that they give you?
In mouse, in mice, you score the size, and in CAM, you count the cells.
Because so my point is that if you are passaging those cells, so at the end you could have cells that are either senescent, so you can have a mix of cells that are capable of proliferating and some that are not. So then you start with the same amount of cells, but they are not the same. So either you just can have cells that are not capable of dividing, but still alive, so senescent, or you just get some cells that are not dormant, but just capable of proliferating, but less fast as the others. So how do you differentiate these possibilities?
So that's not exactly how it works, because so basically what the full model is, the T-HEPs come from a patient you maintain in PDX in chicken. So every week you pass the cells, they make a tumor, you smash it, you put in a new egg, like so. That T-HEPs, they retain their proliferative abilities. When you pass them in the dish, something happens where you select for the cells that are able to be dormant. In the dish, they grow. In 2D, they grow the same. They all grow the same. But it's once you put them back in an animal, like a mouse or an egg, the cells will not be able to grow. But the last piece that you didn't see during the talk is that if you take those DHEPs that are dormant, so if you do tissue staining and you stain for dormancy model, you will see dormancy markers, you will see it accumulate there. But if like in the egg, for instance, you put the DHEP and then for 10 weeks, you pass those DHEPs that you don't see because they don't make a tumor, they just make a small nodule. You keep passing them in the egg, they will reactivate and they will reform a tumor that we call REP for reactivated.
So that was not my question. Actually, my question was, when you reactivate your DHEP, do they proliferate at the same speed as the initial THEP?
Oh, you mean later on?
I mean, either later on or you have
a delay. You have a delay in the grow, but in the mouse, if you wait, at some point you will have them growing back. The work from
so you understand, because my point is that the tumor size can come for two different things. Either you put less cells that are capable of dividing, or you put cells that are capable of dividing, but they divide slower.
So no, they don't divide slower. When you inject them in the mouse, they just don't grow. They sit there and they don't grow for a longer period of time. So that's what allows you this dormant window where you can study dormancy and reactivation.
Because this is super important, because I mean, when you are talking about, you have to distinguish these parameters at the cellular level, because then, I mean, if you just inject the same amount of cells, that's not enough to be able to see, to address what you're actually doing.
I understand what you're saying. And also during the first years, I checked for senescence and I didn't see differences between THEP and DHEP in the number of senescent cells. So this difference between dormancy and senescence, you know, like the dormant state is a state where it's like the cells is able to go back to proliferation, where it's much more like a stem cell. It's much more like a stem cell, basically.
Okay, thank you.
We have a question by Bertrand. Yeah. Go ahead.
Yes, thank you very much for the talk. I was wondering, I hope you did not say it because I missed it, but I was wondering whether it would make a difference for dormancy, whether the metastasis cells travel alone or in small groups.
So that's interesting, because there's the work from Nicolas Aceto in Switzerland, looking at circulating tumor cells. And it's showing that the cells that circulate in groups have better chance to end up in distant sites. But in the early stage of I mean, if you grow the tumor and you sacrifice the mouse 2 weeks after growing the tumors and look at the lungs of these mice in our model, I'm gonna just say like in the HEP model, most of the cells that you find in the lungs are single. So there's maybe at the beginning, there is also this work of early dissemination where the cells is able to leave the organ, like in the case of breast cancer, even prior the tumor development. So maybe at this early stage, it's more of a single cell mechanisms. But when you have the primary tumor established, there is an advantage for a group of cells. But for the dormant state, yeah, early on, it's very, most of the time we have single cells. Yeah, I don't know if I answered your question, but that's...
Yes, you did. Thank you.
Thank you. So I have one regarding the difference between the collagen-free secreted by the fibroblast and the collagen-free secreted by the tumor cells. What it didn't work with the collagen-free secreted by fibroblasts? Do you think it's a different type of collagen? Well, the collagen is different, or is not organized in the same way? Or?
I don't know about that. I know that you always have to be critical, and I'm critical of the paper. And this experiment was the faster we could do. But I would have liked to use the mouse that's depleted for collagen 3. They have a mouse model, but it was in the time frame impossible to get them there because they're frozen at Jackson, so we have to defroze the line and stuff. So, but the other thing is we saw no differences, but it's also a short period of time because in the chicken you cannot keep the cells for long. So I would have liked to do it in mouse and look there if it was really the case, because there is this work from Susan Volk that showed that proliferative cells, if you put them, that's match my data, like if you put the proliferative cells in the mouse that's knocked down for collagen 3, they grow faster. So it's matching there, but she didn't check the dormant state. So that's something that we have been interested, but that I will not need that experiment to give you more details. Truth is that the effect we had when we downregulate the collagen in the dormant cells was enough for the cells to regrow without changing the stroma. So that was also helping us to think that the stroma was not that important, because when you downregulate collagen 3 in the dormant cells, put them in the mouse, the stromas keep producing collagen 3. And that's not enough. That's not enough. So I'm also thinking about the work of Claudia Lobel, where she's working on the matrix that's just secreted, because she's like, the matrix as a cell just secrete, it's the one that's the closest one from the cell. So it's because the cells produce this matrix that's close by, just by convenience bind to it. And if you recall the movie I show you, when the cells are on the layer of collagen 3, they form those small nests. They're like packing the collagen around, and they don't grow in there. So, yeah.
Did you take a sequence between the human and the mouse collagen 3? And I don't know if there could be a difference in the affinity for DDR1 or things like that.
I don't know, because truth is that, when we do the adhesion assay, we do the adhesion with collagen 3 that's from bovine origin, and it binds, so I didn't do that, no, but yeah, I guess the binding site are conserved, but I didn't check that for sure.
I have another question about how do you see the intracellular signaling between the DDR signaling that goes to the dormant states of the cell? I guess.
So you mean DDR1 stat 1?
Yes. No. The interaction, you have the interaction between DDR1 and collagen 3 that will maintain the dormancy states. So how do you think that can happen? How this signaling pathway could happen? In terms
you mean like between DDR1 and the endpoint where the cells?
Exactly. What is the definition of the dormant state? Also, in terms of gene expression, you have, I guess, some.
There's a bunch. there's a bunch of stuff,
so,
but
yeah.
the main point is like you will see an upregulation of phospho-P38, and that can be downstream of DDR1. So dormant cells have a high phospho-P38, low phospho-ERK. That's why because they encounter stress, they upregulate this. So that could be below. We did some work at some point. I mean, the bottom line is like after those years, there was a lot of things we didn't put. So for example, we have, and also at the end, you simplify the paper because it's already like 12 figures, like 6 and 6 supplemental. So you cannot put everything, but we have some work with JAK2 showing that JAK2 can phosphorylate STAT1, activate STAT1, and this through DDR1. We have IP of the two showing together. And there's also this work from the POSI lab where she showed DDR1 in the nucleus. And we have some data where we see DDR1, I mean, at least for part, co-localized with the nucleus. So somewhere, you know, if it's thread, it could go there. What it does, I don't know much, but you know, yeah, I mean, I don't have something more detailed about that, I would say. But yeah, there's a lot of gap in there. Yeah. But I'm glad they took the paper without it. Otherwise, I would still be working on it.
It's already a nice story, yeah. Okay. Other questions from the audience? No? So if not, we can hand it up there. So thanks a lot again, Julie. It was very nice.
Thank you for having me. That was nice.
So if people want to talk to you about,
yeah, I'm still here. If anyone like want to do a postdoc in the US, want to know something about it, you know, just
so
let me know.
Just wait a few minutes if you want.
Yeah, I can wait no more.
Yep.
Salut Julie, à bientôt.
Merci d'être venu. À la prochaine.
So I'm stopping the enregistrement.